Good morning. My name is Claude Jean-Guillaume, and I'm a biochemistry senior here at College. And during the summer of 2013, I was able to work in a lab at Stony Brook University that is looking at how nitric oxide signaling in bacteria can help regulate biofilm formation. And I was able to work on this project involving this newly discovered domain called HOBO, and it was named HOBO by the lab at the time because it's never been studied before and it's never been claimed by any scientific family. So they decided that they would call it HOBO. And the title of my presentation today is Phosphorylation Analysis of HOBO Associated Histidine Kinase in Vibrio cholerae. And HOBO. HOBO is this sensing domain that's been identified in bacterial species such as Vibrio cholerae and Pseudomonas aeruginosa. And HOBO shares many similarities with a very well studied bacterial domain called HNOx, which stands for helium nitric oxide oxygen binding domain. And this HNOx domain, it's been identified and very well studied in many bacterial species. It shares many similarities with HOBO. So I'm going to start to talk about HNOx a little bit, which will help you understand the rest of the presentation about HOBO. So HNOx is this sensing domain that's been identified in many bacterial species. So HNOx is able to bind dimolecular gas species such as nitric oxide, oxygen, carbon monoxide. And what's been found is that in bacteria, binding of nitric oxide to HNOx is able to regulate biofilm formation. And HNOx has been found to be cosistronic with molecules, with protein molecules such as histidine kinase, where you have a signal coming from HNOx signaling your histidine kinase to a signal coming from HNOx signaling your histidine kinase to have to do a phosphoryl transfer to a response regulator molecule down the signaling pathway and to create a certain response in your bacterial cell. And HNOx has also been found to be cosistronic with a dual functioning diguanylate cyclase phosphodiesterase protein. So what this diguanylate cyclase phosphodiesterase protein is able to do is to take two molecules of guanosine triphosphate and to turn them into one molecule of cyclic dimeric guanosine monophosphate. And that same enzyme can take that molecule of cyclic dimeric guanosine monophosphate and turn it into two molecules of phosphoguanosine. And so basically what's been found so far is that signal binding of nitric oxide to HNOx is able to create a signal down a signaling pathway, and this signal is able to create a certain regulation of biofilm formation in many bacterial species. And so basically a bacterial biofilm, is when you have a bunch of bacteria coming together and forming this, they are able to form this polysaccharide matrix, and they are able to assemble in so bacteria are single cellular organisms, and when they form those biofilms, they are able to give the impression of being one multicellular organism, and they are encapsulated in this polysaccharide matrix, and this polysaccharide matrix is able to protect them from their environment. So what's been found is that when you have bacteria in this biofilm formation protected by this polysaccharide matrix, when you decide that you are going to use your everyday antibiotics to eradicate those bacterial infection, it's very hard. It's super hard. It's to actually be able to eradicate those bacterial infections when you start having this biofilm formation, because what the bacteria would do is that once they are in this biofilm formation, they are able to some of the bacteria will get out of this biofilm formation and get into a motile formation where they're able to keep on infecting the organism. And by using your everyday antibiotics, you are able to kill those bacteria in this motile formation. But the main source of the infection, the actual biofilm, persists. So you are not able to eradicate this main source of infection in your organism. And it's been found in many medical cases where patients living with biomedical implants and where you have biofilms forming on those biomedical implants, and it's like a big problem in medicine, where like I just said before, using your everyday antibiotics, you are not able to eradicate these infections because of this biofilm formation. So the interesting thing is that what the lab that I was working in found so far is that nitric oxide signaling is actually able to regulate the amount of biofilm formation you can have in certain bacterial species. So what they found is that when you have nitric oxide binding to HNOx, what you have is so like I said before, HNOx is sometimes cosistronic with dual functioning digranulate cyclase and phosphodiesterase molecule. And what they found was that binding of nitric oxide to HNOx is able to decrease the digranulate cyclase activity of this enzyme and increase the phosphodiesterase activity of this enzyme. So what you have is that when you have binding of nitric oxide to HNOx, it signals this enzyme to decrease the concentration, decrease the kinetics of forming cyclic di-GMP, and increase the kinetics of forming PGPG. And a decrease in the cyclic di-GMP signaling down the pathway is seen to cause a decrease in biofilm formation. So when you have nitric oxide binding to HNOx, you have a decrease in cyclic di-GMP formation, but you do have an increase in phosphodiesterase activity. So you have an increase in phosphoguanosine formation, and you don't have that much signaling, cyclic di-GMP signaling down the pathway. So you don't have that much biofilm formation. So you have a decrease in concentrations of biofilm. So this nitric oxide signaling in HNOx might be able to have many interesting applications in the medical field. And like I said before, this Hobo domain that was discovered in the lab I was working in, it shares many similarities with this HNOx binding domain. And this Hobo domain is able to bind nitric oxide, and it's able to use that signal from nitric oxide to instigate a response in your bacterial cell. But so far, no lab has been investigating this Hobo domain at all. So we don't have that much information about how nitric oxide binding to Hobo, what kind of response that creates in your bacterial cell. If it's able to regulate biofilm formation, if there is a change in gene transcription, no, we don't have any information about it at all. So that's what was very interesting about the project that I was working at the time, is that this new domain, pretty much no lab has studied it before, so we pretty much don't have any information about how nitric oxide signaling in Hobo is able to instigate a response in your bacterial cells. And but what we did find is that this Hobo domain is also cosistronic with histidine kinase that is also involved in two-component signaling. So binding of nitric oxide to Hobo is able to activate the activity of this histidine kinase, and this histidine kinase is able to phosphorylate a molecule down the signaling pathway and instigate a response in the cell. But so far, we don't know what that signal is, no, what that response is, and all the mechanisms involved in this signaling pathway. And so what we also found was that this histidine kinase has a receiver domain on the histidine kinase that is able to accept a phosphate molecule. So this histidine kinase is able to phosphorylate itself. And what we wanted to find, the main purpose of this of the experiment of the project I was working on, was to see how autophosphorylation of this histidine kinase affects the transmission of the signal from nitric oxide, the transmission of the signal down the pathway. So what we found was that the receiver domain for this histidine kinase is able to autophosphorylate an aspartic acid from a phosphate from a histidine residue. So it's able to take that phosphate from this histidine residue and transfer that same phosphate molecule to an aspartic acid residue on the histidine kinase. So in our experimental design, what we decided to do was to mutate this aspartic acid residue to an alanine residue and prevent that histidine kinase from being able to autophosphorylate itself. And we wanted to see if we keep that histidine kinase from autophosphorylate itself, how that would affect the signal down the pathway. And so we create that mutation, and we in our experimental design we had a wild type version of that receiver domain for histidine kinase, and we had a mutated version of an aspartic acid residue to an alanine residue that should not be able to receive a phosphate molecule anymore from that histidine kinase. And in our results, what we found was actually pretty interesting. We found that when we created that mutation, so after we created that mutation in the receiver domain for histidine kinase, we did what is called a 20-minute autokinase reaction, where you incubate your kinase molecule with gamma labeled with phosphorus 32, and so you radioactively labeled the gamma phosphate on an ATP molecule, and you incubate this with your histidine kinase, and you let the reaction happen. And after specific amounts of time, you take a sample from your incubation reaction, and what you do is that you quench it with SDS page buffer solution, and to stop the kinase reaction. And after you stop that kinase reaction, you basically run an SDS page to separate your proteins, and you expose your result to a photographic machine, to a photographic apparatus that is able to produce an image and give you the amount of phosphorylation activity for your kinase reaction. And so basically what we were able to find is that creating that mutation in the receiver domain for your histidine kinase, actually we observed an increase in phosphorylation activity when we did that mutation in the receiver domain of the histidine kinase. So you were able to see on this graph that for the wild type, the signal is kind of pale, but you have a more pronounced signal for your mutated version of the receiver domain of the histidine kinase. So basically, that was what we were able to observe, and it was different from what we hypothesized, because if we think about it, mutating that receiver domain on the histidine kinase and keeping it from being to autophosphorylate itself, it shouldn't be able to have that much phosphorylation activity. So you should be able to see a stronger signal from the wild type than from the mutated type. But this is not what we saw in our experiment. So what we decided to hypothesize from these results is that there might be a mediator protein involved in this case, where you have that histidine kinase being able to phosphorylate that receiver domain, but that same histidine kinase being able to transfer a phosphate to mediator protein, which is then able to phosphorylate response regulator protein down the signaling pathway. And that's one of the explanations that we decided that might explain why you would have a stronger signal in the wild type, where you have that mediator protein being phosphorylated much more, and in the mutated type you have that mediator protein being phosphorylated much more with your radioactively labeled gamma phosphate, and here the signal being split between your receiver domain and that mediator protein, so you have a stronger signal in your mutated version of your receiver domain for your histidine kinase. So that was one of the explanations that we hypothesized that might explain the results that we got in this experiment. And because of the short amount of time that we had to run this project, we were not able to pursue that hypothesis. We were not able to go further with that experiment, so we had to stop with these results that we got for that autokinase reaction. But in the future, this is something that we think might be very interesting to explore, is to see if there is actually a mediator protein that is able to receive that phosphate and to actually regulate that response, that the pathway. That would be an interesting thing to investigate in the future. And thank you for your time. Thank you for listening to me, and I will now receive the questions that you may have for me. Thank you.